Hello and shalom, everyone, and welcome to another session of the Australia-Israel Scholar Seminar Series. Today, we're very excited to have Dr. Dror Minz from the Volcani Institute in Israel joining us. But before we get started with the talk, I'm going to do an acknowledgement of country, which is what we do in Australia to acknowledge the traditional owners of the land on which we are meeting. So today I'd like to acknowledge the traditional owners of the land on which we meet today, who are the Turrbal and Yagara people. I pay my respects to their elders, past, present, and emerging, and extend those respects to any Indigenous Australians joining us on the talk today. So today I'm very excited to welcome Dr. Dror Minz, as I mentioned, and he is the principal investigator in the Minz lab at the Volcani Institute, which is Israel's agricultural research organization. The Minz lab research focuses on microbial ecology and environmental microbiology. So I'm very thrilled to welcome Dror here with his talk called From Microbial Ecology to Application. So I'll hand over to you now, Dror, to give your presentation. Thank you.
Hi, thank you, Joe and Yoni, for inviting me and organizing this session. I'm very happy to be here, at least on the website, being here, not physically, but one day, hopefully, I will visit as well. As I said personally to Yoni and Joe, we have in the group a few projects that are related directly to agriculture, plant growth promotion, disease suppression, and other aspects of microbial agricultural microbial projects. But I chose to show today something that is more environmental and quite exciting. And I'm trying to take the application and show you how we should not be worried about doing ecology. Some people say, oh, ecology is just theoretical and it's not, it doesn't have any implications. And even today, people are using those sentences. So I'll show you how we moved from microbial ecology to real application in reducing nitrous oxide emission from agricultural fields. So I'll start with past. Quite a few years ago, more than 10 years ago, we did a just ecological study looking at the community, microbial community, as we call it now, microbiome, of the roots of different crop plants. So we looked at cucumber, tomato, corn, and wheat, and identified the communities on the root, and we saw some very interesting things. Please don't look at the names of the bacteria. Some of them I don't even recognize myself. It's not relevant. What is important is to see that we have hundreds of populations that are in a very low relative abundance. So to have the right to go on the figure, a microbe should be above 2% of the community as a relative abundance. And you can see that in cucumber, for example, the vast majority of the bacteria are below that. So there's an enormous diversity in cucumber, quite high diversity in tomato. The red arrows are the ones that are unseen because they are in very low relative abundance. And in the two cereals, we have a highly dominant Pseudomonas. If you're not a microbiologist, just ignore it. An organism that is highly dominant, over 60% in maize and 50% in wheat. So that while cucumber is showing high diversity of bacteria in the root, the wheat and corn show high dominance, and this is like two extreme situations. So what we wanted to do then is to choose the two extremes, the cucumber and wheat, and look more deeply into what is the differences in the microbial community. What does the differences mean? So we did what we call metagenome and metatranscriptome. We looked at all the genes of all the bacteria in the wheat root system versus all the genes of all the bacteria in the cucumber system. We're talking about tens to 100,000 of different species in each root. We compare them. We compared the whole genomes of the community, and we compared the whole transcriptome, all the genes that are expressed by the different communities in the two systems. So the question was, do the genes express differently by the different communities and the different plant types? And then jumping into really complicated and impossible to explain, and I'm still trying to understand some of the things here by myself, but what we saw very, very interesting is that if we paint green, if we color green the genes that are expressed highly in the cucumber microbiome versus yellow for the ones expressed highly in the wheat system by the microbiome, by the bacteria in the root system, we see some, while many of the genes are expressed equally or relatively equally in the two systems, some genes and gene groups are expressed very specifically in one versus the other. What we can do, there are many things we can do with this data. For example, we can see in the lower part of the figure, you can see genes that are related to cell wall degradation. So in green, there are the genes that are expressed highly in the cucumber system. And of course, that means that these sugars should be available in the cell wall of the root of the cucumber. Otherwise, the bacteria will not express genes to degrade them. And for example, if you look at the genes that are related to cellulose degradation, you can see that the two systems express different cellulose degrading enzymes. For example, exoglucanase in the cucumber and endoglucanase in the wheat, which suggests that the level of complication in the cellulose, how many side branches the cellulose may have, is different in a different plants. So the bacteria can tell us a lot about the system where they are living in. But there are many, many other things that are interesting. I will only show you a few. I will zoom on a few of them. For example, in red, you can see the square here. These are genes related to dissimilatory nitrate reduction, or denitrification, as we call it in environmental sciences. These are genes involved in anaerobic reduction of nitrate all the way up to N2, dinitrogen gas. So these are expressed highly in the wheat system. And also these genes are also, these are all suggesting to us that there is a lack of oxygen in the root system of the wheat. So the community, instead of dumping the electron into oxygen, they will do it on the on the, why is that? Oh, sorry. Oh, they will reduce the nitrogen compounds. So the conclusion here, we have lack of oxygen. When we look at, why isn't it? When we look deep into this chain of reduction, we can see that we have nitrate all the way to nitrous oxide, and the last gene that is supposed to, the product should take the nitrous oxide and reduce it to dinitrogen, is not expressed in high levels. So this made us ask the question, why, what is going on? Why is the community lacking the option of reducing nitrous oxide to dinitrogen? And then we realized that nitrous oxide emission when we looked at the literature, about 40% of the global nitrous oxide emission is anthropogenic. And out of that, at least in the US, they calculated that 3/4 of the anthropogenic emission comes from agricultural soil management. So this is denitrification, the whole process. The last step is missing in the root systems of the wheat. And we know that nitrous oxide is really the villain in the story because it's a very potent greenhouse gas. It's about 300 times between 265 to 300 times worse than CO2 as a greenhouse gas, but it's also responsible to ozone depletion. So it's a really big thing. And as I said, 3/4 of the anthropogenic emission calculated in the US is from agricultural soil management, and all the others is really minor: transportation, industry, combustion, even manure management. So if it's such a bad compound, and it's a gas, and if this last step is damaged, then nitrous oxide is accumulated in the root system and escaping into the atmosphere. So we thought if this is indeed the case, we should try to solve the problem of the, I don't know, the lack of ability of the microbial community. What you see here is not one organism. This is the total community. So each of these genes that is colored yellow is not by one organism. It's by hundreds to thousands different genes of the same function from different community members. And the community members that are responsible for the NOS-Z reduction of nitrous oxide are either lacking or not expressing the genes in high levels. So we thought to say, how can we minimize the emission? There are basically two ways. One of them is to prevent the production of nitrous oxide, use some antibiotics, some toxic compound that will kill the organisms that produce nitrous oxide, so it will not accumulate and then escape into the atmosphere. This is impossible because there are so many different organisms that can do it, that even if you kill 99.99% of the community members, the ones that are not killed will then take over the niche and continue the emission of nitrous oxide. Then the other option is to consume it before it's emitted from the soil to the atmosphere. And this is something that we took upon ourselves. We basically went into wheat roots, isolated a lot of bacteria under anaerobic conditions in a desiccator, and the only electron acceptor that was available for the bacteria on the agar plates was nitrous oxide. So only bacteria that can reduce nitrous oxide to N2 will grow and form a colony. And we looked at the colonies and screened them one by one for the ability as a pure culture to reduce nitrous oxide. It looks very simple in the figure, and actually it's even much more simple than it looks. This is like really basic microbiology practice, and it was very easy to achieve. And then I'm making a very long story short. We isolated a few that were more promising, and you can see in the y-axis, these are 3 organisms. In the y-axis, you see the nitrous oxide concentration in the atmosphere of these bottles that are closed, and we injected nitrous oxide. We flushed the atmosphere, so there was no oxygen for them. There was nitrate, and then nitrous oxide in the atmosphere. The bacteria consume the nitrous oxide. You see the nitrous oxide going down in all 3 isolates and grew just as if they have oxygen in the system. So they use the nitrous oxide as an electron acceptor that provide the ability to grow to high biomass. So we did the genome sequencing, and we saw that they have the genes that especially important is the NOS-Z. This is the only gene known in the world to be able to reduce nitrous oxide. So it's not a surprise that they have it, because they all show the ability to use nitrous oxide. Okay, and this is another interesting bug. This one doesn't have the first step, so it cannot use nitrate. But if someone else use the nitrate and produce nitrite, it will grow very well. But remember, we are all only interested in this ability, the NOS-Z ability. So then we took the bacteria and do what we call probiotics to minimize the emission. We grew wheat plants, amended the pots with the different bacteria. This is exactly how they look under the microscope. So we inoculated the soil, the healthy soil with all the other bacteria that are there. We are talking about hundreds of thousands of different species of bacteria in the soil, in gram soil, and so we added just one of the organisms in each in high levels to let's say tailor the microbiome, so it will have organisms that are efficiently reducing the nitrous oxide. Then we took the pots, put them in glass jars for specific time, and then we let the system produce the nitrous oxide, and then we took samples of the atmosphere and measured the concentration of the nitrous oxide in the atmosphere. So this is how it looks in real life, and here it's the syringe where we sampled the gas. Usually we did 24 hours, but we could do shorter as well, under light and dark cycles at a constant temperature. We did it in ambient atmosphere that contains some oxygen. So in this case, they will not be forced to reduce nitrous oxide efficiently, because they will not do the denitrification efficiently, because they will prefer to use oxygen. And then we did it also in anaerobic or anoxic conditions, where we flushed the atmosphere using regular dinitrogen. So the atmosphere will be lacking oxygen, will be where we expect to get a push of the denitrification towards producing nitrous oxide as well. When we looked at the two options, when in anaerobic conditions, just one second, I will move your figure. Okay, in the control, where we did not amend with any bacteria, and the numbers here are the number of different pots in the experiment. So we had 81 different pots in different experiments. And this is the level we see over 100 ppm emission on the average in the pots that we did not amend with bacteria. Remember, this is in favor of denitrification, because there's no oxygen. So the level is high. 100 ppm is very high level. This bacteria even produced nitrous oxide by itself, probably. So it's not efficient. This one is slightly efficient, but this one is statistically significant, from 100 to a little over 60. So almost 40% of the emission in the anaerobic condition was decreased by 40%. Okay? If we look at the conditions where we had oxygen, of course, in the control with no bacteria, the level is much lower, because denitrification is not very popular when you have oxygen. It's going to be extremely inefficient to use nitrous oxide or to do denitrification if you have oxygen. So the level is 30-something in the control. And this, again, is not good for us. This one is not good enough. But so 243 was efficient in the anaerobic condition. But bacteria number 14 was highly efficient in the oxy condition. So almost 50% of the emission was reduced. So we can conclude that we have at least two organisms that can minimize the emission of nitrous oxide when it's amended to the soil. soil that has all the other natural community. So this is important. So just 1 second, before I move to the next step, we did real-time PCR studies in which we measured the survival of the bacteria in the soil and on the root. Since the bacteria was isolated from root samples, wheat root samples, we expected them to be successful in colonizing the root and growing, and the root system is exactly where we want it to work. Because the root system is the hotspot for denitrification and production of nitrous oxide, because denitrification is based on the plant exuding available organic matter to support the bacteria in the root system. So in some cases, up to 40% of the organic matter produced by the plant via photosynthesis is released by the plant into the root system to support the proper bacterial community. So if the bacteria have a lot of organic matter, doesn't have enough oxygen, which happens a lot in agricultural systems because of the fact that we have a lot of water that inhibits or at least slows the diffusion of oxygen from the atmosphere to the root system. And we have a lot of nitrate because in agriculture, we have to support with nitrogen, and it doesn't matter which type of nitrogen you apply to your crop, you will always have bacteria that will transform it to nitrate. So whether it's even, if it's ammonia, if it's guano, if it's compost, animal manure, whatever, after mineralization and nitrification, there will be a lot of nitrate. So in the root system, there is nitrate, lack of oxygen, a lot of organic matter, so the denitrification will be promoted. So we saw indeed that the bacteria that we add to the soil disappear within a week or go below detection limit. They cannot compete with the bacteria in the soil, the bacteria and other organisms, nematodes and fungi that will eat the bacteria and consume it, but they do take over the root. So with real-time PCR, we saw that they are dying in the soil system, but they are thriving and taking over the root and doing what we expected them to do. And that's because we did what is expected in ecology. You take an organism from a niche, and the niche was root, you bring it back to the niche, and they make their living in this environment. Okay, so next thing, before we noticed people that want to invest in things that have a potential came and invested, and we started a new company, a startup company. We called it N2OFF, and the bacteria that we used in the lab are now on the way to become a product, and I'll show you the stages where we are now. Joe, can you give me like a 3 minutes warning?
Yeah, so yes, time is almost up. So if you can give us a little info about the company, that would be great. And then we'll ask for questions from the audience.
Okay, we'll do it very fast.
Thanks.
So in all the experiments we did now, we added dead bacteria as a control. So we checked, for example, do we need a lot of bacteria, or we don't need so much? And we realized that it doesn't matter how much we put. We took a suspension, we added 6 ml of it or 3 ml of it, and it's always efficient compared to the dead bacteria. The dead bacteria were there as an organic matter amendment. So yeah, so we have an efficient activity regardless of the amount of bacteria, and then we checked, can we use 1 application of the bacteria or 2 applications of the bacteria? Both worked very well. And then we moved from very small pots to much bigger pots. We didn't have jars for this, so we have to improvise. So we closed it. Oh, skip some. We closed the plants, much more plants, much higher level of soils, and then it worked as good in the bigger pots. And then, so let's jump over that. Okay, and then we checked, is it only working on wheat? Can we do it in other, on maize, for example? Remember, the maize and wheat had a very similar community, and indeed it worked very well. We had over 50% reduced emission from a corn system from a maize. And then we isolated more bacteria, especially looking for bacteria that produce spores, because then we can make a very stable product out of them. And the green ones in this figure are the ones that are producing spores. This is number 14 that I showed you all the data from before. And we now have even much better organisms that can do it much better and also produce spores that can serve as a product. And right now we are in a huge, in Israeli standards, a huge large-scale outdoor experiment. These are very big containers. We carried soils from different places in the country. So we have 4 different chemically and physically different soils and biologically different soils. We applied one of the spore formers, N number 14. We sowed the corn seeds a week ago, and it's already germinating. You don't see it here. So we hope that we'll see the effect of different soils. We know from the greenhouse that different soils behave differently. So this is one step before we move into the field, real agricultural field that will be chosen by after we see the results of those 4 soils. And that's it for now. That's
Thank you so much, Dror. That was really a great talk. Thanks. The virtual round of applause from everyone. I'm sorry, there is a fire alarm that's just gone off in my building, so I hope you can hear me. We just had to evacuate, but I'm here. So could I open the floor up to any questions from Erez or Yoni, just to get us started? You can write in the chat, or you can use speak to the microphone if you wish.
Yeah, Dror, it was a great talk. I'm currently working in Victoria, in agriculture Victoria, and we also concerning with the emission gas, so trying to reduce it, but from a different approach. We're trying to genetically modify the plants, the compounds of the lignin and the fat ratio. So we're talking about the rumen stomach rather than the bacteria, and it was really eye-opening to see how great is the impact that you can create through the bacteria population in the root system. It was great, really interesting.
Yeah, remember I wanted to show that you can go from ecology, from understanding the system, and then identifying a failure or malfunction of the community and try to fix it also in microbial ecology tools or microbiological tools. In a system that if you can affect the emission by playing with plant genetics, so maybe together we'll get a much higher prevention. This is very interesting.
It's really interesting. Yeah, I think the combination of the two will give us double the impact. That's what it seems to be. Yeah.
Yeah. Our next stage after doing the field experiments, or in the field experiments, we want to try to combine this function of the bacteria with chemical inhibition of nitrification, so preventing production of nitrate. If we lower the nitrate in the soil, then there'll be less denitrification. So we're going to play with some chemical compounds that are used by farmers sometimes. Maybe not.
The question between ammonia and nitrate. That's what it.
Yeah, the nitrification is the oxygenating ammonia to nitrite and then to nitrate. And then so if we keep the fertilizers in the ammonia form, that will reduce the emission as well.
Yeah.
Remember, there's no solution currently no solution for nitrous oxide emission. The solutions now is maybe minimize the fertilization, play with the timing of fertilization, things like that, that will reduce the emission a little bit, not very efficiently. So
It's 40% of the agriculture activities are contributing to like nitrate oxide emission. You're talking about reducing by 40%. That's huge impact.
So in some stages we got up to 60%. Of course, one of the things we have to do is play a little more with the conditions we apply, when we apply, how we apply, and maybe improve it or make it constant in different. So it doesn't work the same. All soils, some soils are emitting much more, and then the level of our reduction is better, and some are less. So there's a lot more to do before we can come to the farmers and convince them this. I don't think we have to go to the farmers. I think the farmers have no justification of spending a penny on something like that. They shouldn't, theoretically, they shouldn't care about greenhouse gases from their fields. I think the state should do, the regulators should force it. We did a lot of experiments now also with covering seed coating with the bacteria. It's very preliminary, but we did a lot, and the bacteria work fine. You can just cover the seeds with the bacteria, plant them, and since the bacteria are root organisms, once the seed germinates, the bacteria just move towards the root and take over the root, and they as the root grows, the bacteria also grow. So using the seed as a vector is very promising as a product. So think about a situation where we will not deal with the farmers at all. We just the seed companies will give them the seeds already with the bacteria, and the bacteria are completely safe. We did genome analysis, no toxin, no chance of any damage to whatever. And these are natural bacteria. They're root organisms. You don't bring GMOs. I don't know how it is in Australia. Europe are like sweating when you just say adding anything to the soil.
Similar here.
No GMO, not mutated in any way, not harmful to any organisms that you can think of. Just root organisms. They are naturally there. Even in the controls they are, but at lower level. So.
It's microbiome population engineering, I think you could call this.
Yeah. I like the term tailoring the community.
Yeah, it's a good one as well.
Yeah.
Great. Thank you.
Thank you so much, Erez, for your great question and discussion. It's great to see people working on similar problems on other sides of the globe. I've actually just put a link in here to a research grant. Sorry about the noise. A research grant program between Victoria and Israel that fosters research and innovation collaboration. So that might be something to consider to continue this kind of work. But thank you so much. I will sign off because the fire alarm is still going off.
Oh, wow.
It's really loud. Here, I hope it's not too bad for you. And I'd like to thank you again, Dror, for your fascinating talk. I love the commercialization and the fact something like this could be done to reduce greenhouse gas emissions, globally. It could have a huge impact. So that's really exciting. And thanks for being part of the Australia-Israel Scholar Seminars. And I hope to see you all again at one of our upcoming seminars. So thank you so much.
Thank you, everybody,
Thank you.
for being here. Bye-bye.
Thanks, Dror. Thank you.